Good morning, everyone, and thank you for joining us for today's Forensic Technology Center of Excellence webinar. Today's webinar is on the evolution of SNPs as a forensic marker. My name is Josh Vickers with RTI, and I will be your technical host for today's webinar. We do have a very star-studded cast of presenters today, so I'm going to run through them real quick. Our first speaker today will be Dr. Kenneth Kidd. Dr. Kenneth Kidd is a professor emeritus of genetics and a senior research scientist at Yale University, where he is a human population geneticist. He has published over 550 scientific papers on a variety of subjects before and during his 44-year career at Yale. His research has included medical genetics, gene mapping, database design, pharmacogenetics, and a variety of molecular methodologies. His long-standing interest in human population genetics has been combined with his laboratory's expertise in molecular technology to examine human genome diversity at the DNA level. Our next speaker for today will be Dr. Chris Phillips. He has been in the forensic science field since 1979, starting at the Met Lab, then moving to Bart's Hospital, where he helped develop DNA analysis that was then in its infancy. Since 2001, he has been a full-time researcher in the Forensic Genetics Unit, the University of Santiago de Compostela in Spain. His research has focused primarily on SNP analysis, development of novel forensic markers, which including SNPs, STRs, and indels, forensic ancestry inference, accessing public genome-scale data forensic purposes, and building open-access online population data resources for the genetics community, which is exemplified by SP Smart. Our third presenter for today is Dr. Tom Parsons. Dr. Tom Parsons received his undergraduate degree in physics from the University of Chicago and then his PhD in biochemistry from the University of Washington in 1989. He joined ICMP as Director of Forensic Sciences in 2006 and leads a large team of forensic scientists spanning the disciplines of forensic anthropology, forensic archaeology, DNA profiling, genetic kinship matching, and informatics. This includes the world's largest and most successful missing persons DNA identification laboratory system. He has authored numerous scientific publications and is a prominent member of the international forensic science community, frequently presenting at international scientific conferences and leading workshops on DNA identification of the missing. And our last presenter today, and also our moderator and host, is Dr. Daniele Pedini. Dr. Daniele Pedini is a forensic DNA expert whose current research focuses on microhaplotypes and on the use of next-generation technologies for human identification purposes. His forensic experience ranges from processing crime scenes for biological specimens to processing evidence in the laboratory, and from DNA profiling to testifying in court as an expert witness. Dr. Pedini has been on the faculty of the Department of Forensic Sciences at GW since 2004. I'll run through our quick disclaimer. The Forensic Technology Center of Excellence, these opinions, findings, and conclusions or recommendations expressed in this presentation are those of the presenter and do not necessarily reflect those of the Department of Justice. Any products and manufacturers discussed are presented for informational purposes only and do not constitute product approval or endorsement by the U.S. Department of Justice. All right, before we do get started, I would like to hand things over to our moderator today, Dr. Daniele Pedini, to get us started.
Thank you, Josh, and thank you to all attendees, and a special thanks to our excellent panel of speakers. The thought behind today's webinar is to give attendees an idea of how the concept of SNPs as forensic markers has evolved over the past decade, thanks to the new knowledge that's been generated by the scientific community, and also thanks to the new technologies that have been developed, particularly massively parallel sequencing or next-generation sequencing technologies have been sort of a forensic SNP booster shot. And now SNPs can really complement STRs in individual identification, familial searching, and missing person cases, and can even provide biogeographic ancestry information. So I look forward to today's presentations, and we can get started.
Good morning. As the most ancient of the speakers today, I was assigned the task of looking at the development. And it's interesting to look back into ancient history, a form of ancient DNA, that the first DNA polymorphisms were just beginning around 1980. And when we summarized the number of mapped and known DNA polymorphisms in 89, going back, there were fewer than 1,200. In green are those that were anonymous. We had no idea where they were. Well, we knew what chromosomes, but no idea what they were related to. Only a few here in red are those related to genes. Initially, the DNA polymorphisms were primarily RFLPs, restriction fragment length polymorphisms. They were detected because of sequence variants that altered the length of the DNA fragment that could be detected following electrophoresis. They're now known that most of those were SNPs, single nucleotide polymorphisms, and some of them were insertion deletions. Compared to the 1980s, dbSNP now catalogs millions of SNPs and indels. In forensics, it was 1985 that Alec Jeffreys published his absolutely seminal paper that showed individual identification based on a tandem repeat cord that could detect many highly variable loci, resulting in an individual specific DNA fingerprint that was rapidly incorporated into forensics. My lab got involved a long time ago, finding new RFLPs, working to put the linkage map together, studying RFLPs to map diseases, exploring PCR when it became available, working with haplotypes. I'll get to that more later. My interest in population genetic studies of humans goes back to my doctoral days, and more recently we've established 2 databases that are online, and talk more about those a little later. During the 80s and 90s, there was a transition from those RFLPs into the use of short tandem repeat polymorphisms. Those could be studied using PCR and capillary electrophoresis, and a national database of convicted offenders was established, so that in the late 1990s, there was a standard set of markers that had a clear, defined kit that became commercial kits used during the World Trade Center attack. And you'll hear more from Chris Phillips and Tom Parsons, but SNPs would have resulted in more loci being typable, and SNPs were used, but they were not very informative because at that time there was really insufficient documentation. We learned a lot, and a paper by one of the committees overseeing the identification was published in Science in 2005. The first panels of SNPs for individual identification were published in 2005 and 2006, and these were discussed in a panel report at the International Society of Forensic Genetics meeting in 2007. Dr. Phillips will talk a bit more about that later this morning. Our lab started working on a panel that was discussed at that meeting in 2007 and later expanded, and a paper published a couple of years later. What our SNP panel showed, carefully selecting using our resources to be very uniform in the information for populations around the world, whereas random markers selected because what we knew then were highly polymorphic and primarily studied in Europeans, were not as informative in Africans or Native Americans. We then went on to look at a somewhat larger number, and we had 10 to the minus 30 to 10 to the minus 34 as the random match probability in most populations around the world, except for a few very small, inbred, isolated populations. We could even find that in full siblings, we got random match probabilities of around 10 to the minus 8, 10 to the minus 10, differentiating the siblings. So it became clear that SNPs could provide a lot of information for individual identification, and the field began moving towards ancestry inference. Dr. Phillips presented one of the early papers, and a group of individuals in Michael Selden's lab in California provided another set of markers. In our lab, a study by my wife used Selden's markers and pulled together data on 119 populations, and we collaborated with Carolyn Nievergelt on a panel of markers that she was using for medical genetic studies. But then we moved on towards our own panel, and in a series of papers, we expanded our set of 55 ancestry informative SNPs and finally reached a very large set of 139. And here are structure results for those 139 population samples using these 55 AI SNPs. And it's quite clear that these panels differentiate the sub-Saharan populations, the North African populations, Southwest Asia and Europe, with a climb from south to north in Europe, South Asia into Central Asia. There's not good definitive definition of different groups in East Asia with different colors, but clearly there's a propensity for different frequencies. The Pacific is distinct, and Native Americans are distinct. And just to keep you thinking, other slides that I'm going to show generally go from the left or top, sub-Saharan Africa, through these populations into Native American populations. One of the findings has been that many different research laboratories have published panels of ancestry SNPs, and more are continuing to be published. We looked at 21 different publications a couple of years ago and found that there were only 46 out of 1,300 unique SNPs. Only 46 were common to 3 or more of the 21 populations. What I call a very large empty matrix with different populations studied for different SNPs, making comparison very difficult. However, all of them had African populations, European populations, Central Asian populations, East Asian populations, and the American populations. But when we typed most of those, we could not get better than 5 and possibly a 6th distinction between Southwest Asia and Northern Europe. As we went on, we have added to that empty matrix ourselves by studying a subset of 74 markers focused primarily on East Asia, and we're able to get some distinction between North Asia, East Asia proper, Southeast Asia, the Pacific, the Americas, but also South Asia, Europe, Southwest Asia, North Africa, and Sub-Saharan Africa. So it's possible to continue to improve this. SNP resources are available to the community, and my lab is responsible for ALFRED, the allele frequency database that now has, as of yesterday, over 49,500,000 tables of allele frequencies, free, available online. We've more recently established frog. I like pronounceable algorithms, not algorithms, pronounceable values, forensic research on genetics, knowledge base. It's also freely available. It has 4 panels of II SNPs, 12 panels of AI SNPs that allow calculations. SNPer is something that Dr. Phillips has worked with a lot, and he knows more about it, in fact, than I do. So forensic uses of DNA are worth considering. The individualization II SNPs give random match probabilities, which can easily be 10 to the minus 30, 10 to the minus 50 with a reasonable number of SNPs. Ancestry inference, clearly you're seeing we're beginning to get biogeographic regions around the world clearly distinguished. Familial relationships are reasonable with STRs, but much more difficult with individual SNPs. There are many individual SNPs now available for phenotype inference, eye color, hair color, skin color, and increasingly mixture deconvolution is becoming a major issue in forensic practice. It's difficult to do well with the short tandem repeats. However, the bottom line here is that microhaplotypes can be good for all of these forensic purposes. So what's a microhaplotype? A segment of 2 or more SNPs, could be an indel, but I'm dealing in my lab only with SNPs, within a short distance from each other, 300 nucleotides maximum, with 3 or more allelic combinations. And the short distance allows the brand new, at least in my career, massively parallel sequencing to read across the entire difference, resolving phase, thereby separating the maternal and paternal chromosomes. And here is a Sanger sequence of a small coding segment of HLA-DQ-A1. And 2 of these SNPs result in amino acid changes. 2 of them are silent. But keep this in mind. I'm using this as an example throughout here. So to reprise, microhap is SNP-based, multi-allelic locus, small enough amplicon for a single read with massively parallel sequencing. No stutter, a common problem with short tandem repeats, even using massively parallel sequencing, and all alleles are the same size, a problem that confounds massively STR loci, and there's a very low mutation rate. So here's the example again. There are 16 different haplotypes that can exist for these 4 SNP sites and 136 different genotypes. So the question is, what's the genotype of this individual? It does make a difference. So if we look at the 16 different haplotypes that are possible, there are 4 different ways one can be heterozygous at all 4 sites, and these have the same unphased phenotype. Using Sanger sequencing, they are heterozygous at all 4 sites. However, all 4 have different pairs of the 4 possible proteins that can be synthesized. Hence, these are potentially clinically and certainly very different at the actual protein level. It does make a difference. So massively parallel sequencing allows the 2 chromosomes of an individual to be separated and sequenced independently. So I became interested, and my lab got involved back in 2012 in what I'm going to call the exploratory and discovery phases. And early this year, we published a paper on 130 microhaploci evaluated on a set of 83 populations. All of these have subsequently been expanded. We're now at 182 loci. All are less than 300 base pairs, nearly half less than 100 base pairs. Hence, a lot of them can be very useful on degraded DNA, as Dr. Parsons will discuss later. We have typed them and phased them statistically, which is not very inaccurate. It's quite accurate at the population level and accurate with some degree of uncertainty at the individual level. At the individual genotype level, it is not sufficiently accurate for forensic use. And we've put all of those allele frequencies for the first 130 and plus some additional ones in Alfred with the keyword microhaplotype, so they're easy for individuals to find. So let me give you some examples. 65 of them we've expanded to 96 population samples involving nearly 12,000 individuals typed for 469 SNPs. So these frequencies are estimated by phase, but remember, as a statistic with this large number of individuals, it's very accurate at the population level. So here, each row across represents the average haplotype frequency in that population. And they're simply grouped here by biogeographic region. This is a microhaplotype on chromosome 1, number 205 in our lab, KK. 154 base pairs involving 4 SNPs. And out of those 4 SNPs, we identify 5 clear haplotypes and some that were not able to come up with a reasonable frequency that we've lumped as residual here. This has an effective number of alleles that ranks number 12 in this set of 65 microhaps. That's good for individual identification, family studies, etc. The informativeness, it ranks 62. It provides very little biogeographic differentiation. There are 5 common haplotypes virtually everywhere in the world. Here is one that is only 78 base pairs and ranks 10th for informativeness. Notice that Africa and East Asia have a very high frequency of the haplotype GG, which is virtually absent or very rare in Europe, Southwest Asia, South Asia, and even Central Asia, and especially essentially absent in the Americas. So it ranks fairly high, 10th in informativeness for ancestry. The third one is the best for, of the 65, for the number of alleles, the effective number of alleles, and pretty high for informativeness around the world. Notice green and orange in Africa, less blue in East Asia. But notice almost all possible haplotypes appear to be present. This is one that I have to say we need accurate sequencing of to determine whether all of these haplotypes really evolve. Is this a high recombination area? This would certainly indicate that at least historically there was a very high recombination rate, and yet it's not a very large molecular distance. And one last one that we're just starting to work on, 2 SNPs, only 11 base pairs apart, and notice that this population up here in Southwest Asia is the Samaritans, and these are all South Asia populations, India and Pakistan. This is a highly restricted common variant, and East Asia has still a different variant, very common. So it's possible to get quite a bit of ancestry information for some parts of the world. And here, for those 65 microhaps on 96 populations, are structure results at 7 forced categories and at 10 forced categories. At 7, we've got pretty good resolution in geographic regions as noted at the top. At 10, we begin to see differentiation in terms of East African and pygmies have some different frequencies than do Central and West Africans. Europe is a kind of mess divided among three of the clusters. But North Asia and Central Asia are separating out. These are Mongolians and some Siberians. And the 1,000 genomes Americans are clearly highly admixed compared to the samples we've collected over the years. The PCA also shows divisions of geographic regions, and clearly the most distinguished on 1 versus 2 are the Africans, and here the East Asians. The Native Americans are separated. Here are South Asians, and here are Europeans and Southwest Asians. If we go to 1 versus 3, we quickly see that we get very different separation for the Native Americans. And here are the 3,000 genomes: Colombians, Mexican Americans from Los Angeles, and Peruvian individuals. So, very interested in the question of mixtures. And we showed that 2 values, the effective number of alleles, which I showed in ranking, and the informativeness among populations, were good statistics to use. And here, for the 130 microhaplotypes in our paper earlier this year, one doesn't know how good a locus will be until one evaluates it, and there are some that are very good by both criteria, and there are some that are really not very good by both criteria and will not be followed up on. So we're now ready to consider the transition to an evaluation phase. And we've been collaborating with researchers at Thermo Fisher and with Daniele Podini's group at the George Washington University. And they are doing, with some of my consulting but no lab work, identifying from the total of 130 plus a few newer ones, the best ones, with a focus on ancestry, with a focus on the effective number of alleles and their function with massively parallel sequencing, resulting in a panel of 74 microhaplotypes. And here's an example of 36 of those microhaplotypes evaluated with number of reads. And I'm going to focus in on this one, where there are a total of 6 different haplotypes with a significant number of reads. So there are clearly 5 alleles that belong to minor contributors. And though 219 and 192 are very low on this, that means the sequence had that combination of SNP alleles in roughly 200 of the several reads, meaning it's far more than chance would allow. But this is the nature of the mixture that was done, with 10 times more of the homozygous major contributor and a tenth of that for each of the single locus contributors, one of which was a homozygote. And that results in the mixture that was seen. So it does work. I'm not going to really talk about the STR polymorphisms, but they don't work as well with massively parallel sequencing in mixtures because stutter peaks confound interpretation. In our original 130, we did a theoretical number. We had 28 with the effective number of alleles worldwide greater than 3, and the probability of not seeing a third allele was the product for 21 that were at 3, 5 at 4, 2 at 5 effective alleles. Hence, the probability of missing a second contributor is less than 10 to the minus 7. Question is, here in these 36, it was clear that every one of the 36 identified a minor contributor, but how many minor contributors were there? Well, only one identified 4 individuals, 2 at least 3, 15 at least 2. And if we look at these, we can see that they are different and different numbers of individuals. Here, there are at least 2, but the unequal number of reads for TG and GG suggests maybe a third. But notice that all of these sequences were done on the same DNA sample, read simultaneously by massively parallel sequencing, and then assembled electronically by locus. So they all must have at least 5, at least 4 contributors. The question is, how do we determine what that number is? And one bit of logic is self-evident that the more individuals in a mix, the more likely that more of the possible alleles will appear. And here is a simulation of a mixture with 36 different loci, and you can see that anything above this bar labeled 3 will be in this green distribution and with near certainty. Anything above the bar at 4 will be possibly only 3, but possibly as many as 5, similarly. But if we look at some real data, here are 3, 4, 5, and 6 locus mixtures, the total number of alleles actually identified goes up with the number of individuals in the mixture. And if we look now to 78 microhaplotypes, and remembering the panel we're working with now is 74, clearly that expands this distribution, making number of microhaplotypes easy to detect. Let me close by talking about acceptance in the court. Will microhaplotypes meet the Dobert and Fry criteria depending upon the jurisdiction related to new types of scientific evidence? Summarized, they want to know whether theory and technique employed is generally accepted in the scientific community, whether it's been subjected to peer review and publication, whether it can be and has been tested and is reliable, whether the known or potential rate of error is acceptable, whether the research was conducted independent of the specific ligation. And in those, there's also the question that is the laboratory methodology meeting those criteria? Are the statistical interpretations meeting those criteria? Those are two different components of DNA in the courtroom, and both need to be documented. The problem as of today is that my lab and the collaborators I've mentioned are the only ones that have really published on microhaplotypes to any extent. But that is changing rapidly, and I know that there are papers in press by other groups, and as you'll hear later today, in the works right now. There are precedents for all of these aspects that have been accepted, and even SNPs were accepted decades ago, something most of you will not remember, called polymarker. And clearly, sequencing of DNA has been accepted by the courts. So I think it will be easy. I conclude that microhaplotypes are the next generation of forensic DNA marker, but they lack the Linus blanket I refer to as the reference database of convicted offenders. They fulfill all the criteria for a multi-locus forensic panel, including missing persons identification, as you'll hear later. And I think they're much better than the short tandem repeat polymorphisms for many different reasons. The read ratios are comparable to STRP's peak height ratios. The contributor read ratios in the mixture are consistent, or certainly appear to be throughout loci. They allow identification of multiple contributors. They're effective on synthetic mixtures, and as you'll hear from Dr. Podini later, on clean, not clean, but obvious forensic samples. And there's no stutter, the confounding factor in STRPs. So to acknowledge again my group at Yale, the Thermo Fisher group and the group at George Washington University. And I want to acknowledge that hundreds, indeed thousands of individuals around the world have volunteered to give DNA samples that allow these kinds of studies to be calibrated, conducted and calibrated globally. And I thank you for your attention. And here is my lab's website and the URLs for the
Thank you very much, Ken. That was great.
Thank you.
The next speaker will be Chris Phillips.
Hi everybody. Just wait for my presentation to load. Takes a while. And I want to thank Danielle for setting up this webinar. It's good to be part of it, and thanks to everybody for either getting up very early in the morning or staying late at night or somewhere in between. So I'm going to talk a little bit more about microhaplotypes and SNPs, but following Ken's excellent historical overview, just adding a little bit of detail, no more than that. So I want to say a little bit about why we want to look at SNPs in forensics in the first place, how microhaplotype genotyping has become possible with the introduction of massively parallel sequencing systems in forensics. I wanted to add to Ken and Danielle's overview of mixture analysis with the Thermo Fisher mixture analysis panel, with our own experiences of looking at mixtures. It's early days for us, but really wanted to give you a feel for what kind of patterns we're getting. They're really complementary to what Ken has just outlined. And this is using a custom microhaplotype panel for ancestry purposes, but it illustrates quite well what might be possible with mixture analysis of microhaplotypes. And then I'm just going to finish with an introduction to some of the work we've done to develop a missing persons panel, and Tom will pick up that baton and fill in a lot more detailed information about how that panel's developed, which includes microhaplotypes and is a move away from binary SNP markers. So why do we look at SNPs in the first place in forensics? We've got STRs that are well established and populate national DNA databases at great expense and effort, of course. So SNPs are largely going to be complementary to STRs in forensic use, and this is because it's possible to amplify very short fragments that contain the SNP. In theory, we can look at fragments that are just 41 nucleotides in length with the 2 primers and the SNP in the middle. So these are likely to be good bets for very degraded DNA where the DNA is progressively shortened. And of course, SNPs are specialized markers for beginning to know something about the genetic variation that a donor might have. So that can include their externally visible characteristics and their ancestry. And a lot of SNPs underlie, coding SNPs underlie visible characteristics, of course, as well as promoting SNPs, promoter SNPs. And a lot of SNPs are used in ancestry informative tests for forensic purposes because they show good differentiation between populations. And as Ken mentioned, this was discussed in detail at an ISFG meeting in Copenhagen some 10 years ago now. And I think it was Ken that proposed that SNPs can be classified into 4 main groups: identity testing SNPs, those are the SNPs that are going to be short and complement STRs for very degraded DNA, ancestry informative SNPs, trait predictive SNPs, the coding SNPs that underlie common traits, and lineage SNPs. I'm not sure if mentioning lineage SNPs was anticipating microhaplotypes. Ken will be able to say later on if that was true or not. I think he probably obviously had an inkling that microhaplotypes are very informative for lineages, and that's how they were described at that time. And these ideas of classifying SNPs for purpose was carried further in a nice review by Bruce Bedoli and Angela Van Dahl in Biotechniques the following year. And as we've heard, SNPs in closely linked sets can be jointly genotyped when we apply massively parallel sequencing techniques. So this allows us to phase the microhaplotypes when they have multiple alleles, and this increases the discrimination power of short sequence segments markedly, because we're looking at a whole range of alleles rather than just two alleles. I'd add two other classes of SNPs since the panel discussion. Indels, we've looked at indels in detail because they provide a number of properties that are complementary to normal binary SNPs. And we're also able to look at SNPs in and around STRs now with the advent of MPS. So dbSNP classifies all of these markers as short genetic variants, SNPs, indels, and STRs as well. And we can type phased microhaplotypes with MPS and SNPs in and around STRs with MPS, of course. So we can begin to home in on SNP variation within a repeat region, which wasn't possible with length-based techniques, and we can begin to assemble SNPs in phased strands. So going on to this idea that short fragments are good for degraded DNA, as Ken mentioned, an early application of SNP typing was the 9/11 victim identification program, the Twin Towers attack, and it was very early in the use of SNP typing, and so it was very much a pilot. Both technologies were using a system developed by ORCID called SNPit, and they were genotyping very small-scale SNP multiplexes. So Ken's right to say that there was very little reference data available, so the statistics were somewhat compromised, but also the multiplexes were not completely optimized. So although they were small and they contained SNPs that were amplified from very small fragments, the quality of the results was not as good as what might have been anticipated, and very few extra identifications followed from use of SNPs. In our lab, we've been looking at missing person identification or identification of skeletal remains using SNPs and using the snapshot technique. So the advantage of snapshot is you can use optimized capillary electrophoresis machines that are in place for STR analysis and mitochondrial sequencing. So you hit the ground running in terms of the sensitivity of the technique, and it's a nice simple system to apply. And it gives successful results with very degraded DNA. So we can get near full profiles when we're looking at very degraded DNA. And the findings that we get are that short amplicons do give the best possible results, even when the general DNA concentration quantitations are low. So in the chart that I'm showing here, these are indels, but the same rules apply to indels. They're short, they work well, and this is 46 combination of indels, and the majority of them give full profiles for very degraded DNA. And there is some linkage to the amount of DNA that's used in the reaction, of course. Moving on to MPS, well, we used an interesting pilot study for MPS. We were able to use some DNA extracted from a skeleton from a medieval burial site. So this did contain skeletons from the 5th and 6th century, but we looked at DNA taken from the femur of a 12th or 13th century skeleton from the Innsbruck lab. And I just realized that the chart here on the left-hand side is inverted for some reason. I don't quite know why that is, but that's probably a keynote to PowerPoint transition. So the widest bar should be at the top, and the narrowest bar should be at the bottom. So these should be ranked in order of coverage. So the top coverage SNPs should be at the top here. So that was unanticipated. So if you can imagine this chart inverted, so the widest bars are at the top. What we did here was we could see that over half of the SNPs we analysed in this 169 SNP panel gave coverage more than 100 times sequence coverage. So that's a very good result, more than we expected. And more than another third of the SNPs gave coverage more than 20 times sequence coverage. So that just left the SNPs at the bottom here, which were either not giving any results or the quality was poor. And there's a kind of a match, again, imagining this inverted, there's a kind of a match to some extent to the length of the amplicons. So I put these roughly into groups of increasing amplicon size, and there's quite a good match. In addition, the panel that we used from Thermo Fisher has been modified for commercial release, and some SNPs have been taken out, and a third of the SNPs, well, 60 of the SNPs out of 169 have been shortened. So that means that the performance is likely to be even better using this commercial kit for very degraded DNA going forward. And in reducing the panel, they've also reduced the size so that the performance is better. And the same applies to Illumina. Illumina take the option to include SNPs and STRs together in their MPS kit for identification purposes. So you get the opportunity to match the STR results to database entries, and at the same time have the option to look at very degraded DNA and perhaps pick up more results from the SNPs, which are the blue diamonds here, compared to the length of the STRs that might not amplify up so efficiently with degraded DNA, which are the pink bars here. So you can see that the majority of SNPs in the Illumina panel have amplicon sizes below 126 base pairs. So they're likely to be good bets for very degraded DNA. And it should be no surprise that the lab that's responsible for additional identifications in the 9/11 identification program have gone back with MPS to successfully pick up some additional amplifications. This was a report that was published in August of this year, so MPS is being reapplied to previous programs, identification programs, where the DNA is extremely challenging, of course. So I'm just going to very briefly, because I was aware of Ken's slides covering the structure and features of microhaplotypes very well, just very briefly reiterate a lot of what he's said. So Ken, of course, was the champion of microhaplotypes in the first place, and it's very timely that these have been discovered and compiled, because MPS is the ideal system for typing microhaplotypes, and knowledge of phase is critical for forensic use of microhaplotypes. So it's only when you can sequence the whole strand and the SNPs as a single sequence that you know the phase, and you can properly compile the haplotypes in any one microhaplotype locus. So again, I've used the example in this case of a 3-SNP haplotype, microhaplotype, that Ken followed, that we can have 3 heterozygotes, and there's no knowledge of the way these are arranged on the maternal or the paternal strand. If we just do normal SNP typing of these 3 individually, but if we can look at the strands, then we can unlock a whole series of different combinations. It's fair to say that most of the theoretical haplotypes that are possible with a combination of any one combination of SNPs are not seen. Generally, it's a subset of the haplotypes, and this is likely to relate to the way haplotypes evolved. So it's not it's a sequential set of SNP events, I guess. So, for instance, the GTC is just seen as the G allele here in one type of haplotype. So it's likely that the G was a historical SNP event that followed on from the SNPs that are already in place, the background, if you like, and that's why we just see the GTC combination. It's tempting to say the pink TCT is perhaps a very rare recombination event, but this is just speculation on my part. So that's just a reiteration, really, of what Ken explained about the structure and features of microhaplotypes, and the fact that it's possible to type them with MPS successfully. So I'd just like to say a little bit about some of our experiences with mixture analysis. They're not so extensive as Daniele and Ken's slides, but they just give you a feel for how we've adapted mixture analysis with using the MPS analysis software that we use in Santiago. Now, before MPS, we were really challenged with mixtures. So mixtures are a predominant feature of all forensic DNA analysis, of course, and STR analysis is developed very much with the view that more loci are better because they give you more opportunity to see multiple alleles in a profile. With SNPs, that's not possible because they're binary, predominantly binary. So we started to introduce triallelic SNPs for mixtures, but you're stuck with the technique. So snapshot is very much anchored to the dyes that are used, and the dyes, different dyes for different alleles means that you get very different peak heights within any one marker, within any one SNP marker. So the snapshot profile I've got here shows a wide range of peak heights within any one marker, and this is unmixed DNA. This is just normal DNA, normal control DNA. So our answer to this was to use indels, because with indels you can use the same dye within any one indel. So you get much more balanced peak heights. So here's two examples of the same blue dye being used within any one indel, and you get nice balance. And this means that when you do look at mixed DNA, this is a simple two-person mixture, but it illustrates the point that you get skewed peak heights just like you do with STRs, and you can begin to see that there's a minor component and a major component. It's not as quantitative as we would like it to be, but it's an indication. And of course, this is something you can use with very degraded DNA because indels, like SNPs, are based on very short amplicons. So it's a solution to a problem. It's a simple solution, and it's not always effective, but it was our go-to solution for mixed DNA prior to MPS. So since MPS has arrived, we've been very interested in looking at mixtures and to see to what extent mixtures can be properly deconvoluted on the basis that the sequence ratios, or the number of sequences that you see, match more or less the peak height ratios that you would get in a normal STR profile. So there's a direct relationship between the sequence ratios and the input DNA if it's mixed. So I'm going to use, to illustrate the work we've done with mixtures, a new panel that we've been developing for ancestry. I'm not going to say much about ancestry analysis with MPS because, again, that's been covered by Ken, and I know Danielle is going to say a little bit about that later on. But anyway, the point is that this is a panel that's come out of an existing AIMS panel for MPS, which we call Global AIMS. So it was published 5 years ago and was based very much on a genomics panel called the LACE panel, which was a very extensive set of ancestry markers. It obviously factored in some of the 55 from Ken and some of the 128 from Michael Seldin's lab, as previously mentioned. So this new panel is an evolution of the original Global AIMS panel. It includes Eurasiaplex SNPs, which are informative for South Asian and Middle East populations. It includes a larger number of triallelic and tetraallelic SNPs, which we published on recently for mixture analysis, and it's based on a core of the best of the original Global AIMS. So it's got 145 single-site SNPs. And we've supplemented this with 20 to 22 microhaplotypes. I say 20 to 22 because 2 don't work well or sufficiently well in MPS, and that's an interesting observation for us. So you can choose markers for MPS, but there's no guarantee that they will work within your multiplex or within your particular platform. So we've rejected 2 microhaplotypes on the basis of their performance. Anyway, there are 20 microhaplotypes in this panel as well. So when we look at some artificial mixtures, we've combined controlled DNAs from Coriell at 1 to 1, 1 to 3, and 1 to 9. So we started with very simple 2-component mixtures. We get a variety of patterns, and we're using here a prototype plugin for Genotyper. This is the Thermo Fisher panel. So we're using Thermo Fisher S5, the IM PGM S5 to detect these microhaplotypes. We get these patterns. So the plugin is called Microhaplotyper version 8, TDC Microhaplotyper version 8, and it gives you the number of alleles that are observed, you have the opportunity to go to Integrated Genome Viewer to see the pattern of sequences obtained, and it gives you the filtered alleles. By filtered alleles, these alleles that have been recognized as alleles in the system, not misincorporation or sequences that might be false and therefore at a very low level of sequence reads. So it does a filtering step for you, and our scientist Maria has had a lot of work to filter out misincorporation or false alleles in this respect. So we just look at a few examples. They pattern quite well. So when we've got 4 different haplotype alleles between these 2 donors here, you can see it 1-to-1. The read ratios are quite good and pattern quite nicely as we go to a lower level of this P1 component here. There's some imbalance, and this can sometimes be quite marked. So that factors into the interpretation of the patterns. So I would challenge the idea that this is fully quantitative, because the read ratios in normal heterozygotes do differ, and so that's really a factor that should be considered when we're looking at mixtures. And it's by no means the case that you get perfect balance in heterozygotes, even in normal DNA. So here's 2 examples where we've got the dual problem of allele sharing between the 2 components. These are 2 microhaplotypes that are less polymorphic, so that should be no surprise. And we've also got the problem of imbalance. So in this first example here, we've got a unique allele in P1, but it's very imbalanced. So the 1-to-1 mixture really looks imbalanced in the opposite way. And it's difficult, therefore, to be quantitative about the contribution of both components here. Here we've got full allele sharing, so these are uninformative, as you would expect. So it really does depend on the balance that we see between the sequence read ratios in an MPS situation. And we've taken this further. So before we started to look at microhaplotypes and mixtures, we were interested to see if normal binary SNPs would provide some kind of indication. When they're used in large numbers, it is possible to do this. And this recently published paper where Øyvind Blecker and Peter Gill in Norway did a lot of work on some output we produced from MPS analysis using binary SNPs. And this is the 169 SNP panel that I showed for the skeletal analysis from Boulders. And when we look at likelihood, composite likelihood ratios composed of the 2-person mixtures at these ratios, then we do get positive figures compared to the negative figures for the non-contributor hypothesis, the P3 hypothesis. This is based on 100 simulations, so we can look at the distribution of quantiles within the likelihood simulations. And furthermore, we can look qualitatively, so we can look at the presence and absence of alleles, or we can actually go further and look at, to some extent, quantitative analysis, so we can actually look at the ratios that we see between the read densities for each of the alleles. So these are binary systems, these are less informative microhaplotypes. It's still possible with a large number of markers to say something concrete in simple mixtures. So I'm going to finish just as a handover to Tom about the work we've been doing with ICMP to compile more informative markers, specifically for a new missing persons panel. And as I said, this is really a transition from binary SNPs, which are short but a lot less informative than microhaplotypes, and tri- and tetraallelic SNPs. So what we did here was we tried as much as possible to base the panel exclusively on multiple allele SNPs. We don't have any problems with mixtures because it's a very large set of markers in this multiplex, and we have much more information per marker because we're looking at multiple alleles. So the kinship analysis, which is obviously what you're doing with missing persons analysis, you're comparing to surviving relatives, that extra information per marker really makes a difference, as Tom will show. So the ICM panel was a collaboration between 5 labs, and as I said, it combines a very large number of single-site SNPs and the microhaplotype loci. So Ken very kindly provided the lists or the details of these microhaplotypes just ahead of publication. We're involved in selecting the best of those for this purpose and combining those with the triallelics. QIAGEN was the chosen NPS chemistry, and we've already worked very closely with Andreas Tillmar in Sweden, who's the expert at simulating kinship test likelihood analyses and looking at linkage. So ourselves and Andreas had already been familiar with the QIAGEN NPS technology, so we were evaluating a 140 SNP identification panel. This is not using QIAGEN's NPS chemistry known as QIAseq. This is just a PCR and library preparation kit that can feed into existing systems. So this can be used for the MySeq or the IMPGM. And Andreas developed this, was instrumental in developing this panel for forensic use and tested it for the MySeq system, and we did the same for the IMPGM. The 140 SNPs are based on the SNP for ID 52 and Ken's additional 90 or so, I think it is. So the results were good. This is using a third-party kit, as I said, directly into, in this case, with this chart, the IMPGM. So we've got a nice spread of coverage and good level coverage with our Coriell control DNAs. Very few SNPs gave coverage levels below 100 or between 20 and 200. So we were pleased with the results. And when we extend this to a dilution series and degraded DNA, in this the bottom two profiles, this is the femur extract for a very degraded sample. We were pleased with the results, so this looks good. But while we were doing this, the QIAseq chemistry came online, and this offers the prospect of much larger PCR multiplexes. So we were really looking to be able to build a much larger panel than hitherto. And it uses a nested primer system, which Tom will outline. So there's also the prospect for enhanced sensitivity. So of course we were very interested in being able to build a panel for this chemistry. It's also worth mentioning that with the prospect of larger multiplexes, you have to take linkage into account much more carefully. So obviously as the number of markers per chromosome builds with large multiplexes, it's important to begin to factor in linkage, physical linkage, when you're looking at related individuals and adapting the likelihood calculations that you make to establish or to refute a kinship claim. So for this reason, we'd already been working quite closely with Andreas on a system called ELIA, which is an R package which will work with Familias for kinship analyses. And Andreas used the recombination rate calculations we'd already made for STRs, and we expanded for SNPs using the HapMap recombination map, which was published some years ago. So this is timely as well. It's important to factor in linkage analysis when looking at linked loci. And you can see here we would just look at 20 STRs and 52 SNPs. That it does affect the likelihood calculations, particularly when comparing related and unrelated individuals. So it's important to, as I say, to take account of linkage. So with the shared multi-habitat data from Ken, we were able to choose the most polymorphic or the most heterozygous multi-habitats to include in this panel. So we selected 46. These have high heterozygosity values. In most, we use the 1,000 genomes population data because all their data is phased. And Tom really wanted to have universal informativeness. Obviously, ICMPs reaches across the world, so it's important that they're equally informative for Africans, Europeans, East Asians, etc. So that was a bit of an ask, but you know, it was important to establish that we had a reasonably balanced set of heterozygosities levels across population groups. And you could see from Ken's slides that that's not always the case with some microhaplotypes. And going back to what we were saying about the importance of amplicon size at DNA size, we did actually decide to reduce the size of some of the microhaplotypes. So there's some very informative ones which were quite long. So we decided to trade a reasonable reduction in informativeness, as I've said, against efficiency with degraded DNA. So we felt that that was important. And because we were looking at potentially a multiplex much bigger than previously, we decided to also to look in detail at a very large catalogue of triallelics. So obviously, well, perhaps it's not so obvious, but it's clear that this type of triallelic pattern where you've got an even spread of alleles is the best kind of triallelic marker. It's obviously going to be much more informative than the best binary, and these are where the green allele is less frequent, potentially per locus, not so informative. But what an interesting thing resulted from one of Andreas's simulations, which was to look at a whole set of perfect triallelic SNPs, 300 perfect triallelic SNPs, half perfect and half one allele being very rare compared to the other two, or rarer than the other two, and then a panel which was exclusively made up of almost binary SNPs with a rare third allele. And in fact, you can see that the shift in likelihood projections or distribution of likelihoods is quite marginal. So it's interesting that when you do actually include some triallelic STRs that are supposedly less informative, it doesn't make a big difference to the actual kinship analyses. Here's some examples of the microhaplotypes that we did actually choose. These have heterozygosities ranging from 0.68 to 0.73, higher in fact than 0.73. So these potentially will be very informative in routine use and have a very large number of haplotype alleles for three SNP haplotypes. So these are going to be very informative in the long term. Likewise with triallelics, these all have average heterozygosities between 0.6 and 0.66. There are plenty to choose from. They're not always universally informative. So you can see that the Americans tend to have less polymorphism than other population groups because of their population genetics history. But generally, they're quite comparable across population groups. So it's good to include a large number of these. In fact, it was a very large number. So in the end, we were able to compile 1,400 triallelic SNPs, all with high average heterozygosities. This is how they look when heterozygosities are ranked. So the majority have very high heterozygosities, and only a very small proportion, 1.5%, have heterozygosities below an optimum binary SNP heterozygosity of 0.5 or 50%. And these are ones that have very skewed allele frequencies. So these are average values, and some of the population groups have pulled down the average heterozygosities in this case. When we include on the same chart the microhaplotypes, you can see that a very large proportion of the microhaplotypes here have even higher informativeness. So 39% of microhaplotypes are well above the 0.66 maximum for triallelics. So again, we're going to get good information for kinship analyses from this type of SNPs. This one here is a microhaplotype with a triallelic SNP. So it's a nice combination of both types of marker, if you like. So that has a much higher heterozygosity as a result. So with the size reduction, we were careful to try and preserve as much as possible of the informativeness, but we decided that it was really a good idea to actually try and reduce the span. So that's the distance between the two outlying SNPs as much as possible, because we knew that the primers were not likely to be completely close to the microhaplotype span and be some distance apart. So the actual amplicon size could potentially be much longer than maybe 150 to 200 base pairs or nucleotides. So we really wanted to make sure that we could avoid very, very long amplified fragments. So this is really how the size reductions looked. Okay, that's it. So I'm just going to finish with just one final word of caution that's come out in the last 3 months, and that's that about 10%, or 7%, Tom said, but it's likely to be a little bit higher, between 7 and 10% of the triallelic sites that we put into this panel are actually segmental duplications. So we knew already that a very large proportion of the triallelic sites that we've compiled from 1,000 genomes are not actually true SNPs. They're artifactual as a result of segmental duplications, and that's been the case. So this is really just, I guess, a failure of the bioinformatics pipeline that goes into constructing these multiplexes. And when you've got a very large number of markers going in, that's understandable, I guess. So anyway, that's just a word of caution about assuming that all markers are going to work perfectly and be true markers, of course. And you know, this is just this problem that's well known of comparing allelic diversities against apparent diversity from segmental duplication. So in a diploid chromosome pair, this is likely to be a common occurrence. It doesn't happen in the Y chromosome, but duplication obviously is a common feature of many Y STRs, particularly the rapidly mutating Y STRs. So shouldn't be surprised that segmental duplication is a dominant feature of the genome, and to be wary of it. Okay, that's it from me. I'd like to thank all of you for your attention, but primarily like to thank the group in Santiago who've helped a lot over the years with both SNP analysis and microhaplotype analysis. In particular, I can use the pointer again, Maria de la Puente, who's done a lot of work recently on optimizing the genotyper module that we saw for microhaplotyper. So looking at microhaplotypes in mixtures, so kudos to her. Fonde, Carla, Anna, and other members of the team have all been very helpful in compiling SNP sets over the years, particularly for ancestry and for identification. And I should also mention that Vicky Lariou is the principal investigator for all the studies I've outlined and head of department, and Ankol too has taken a lot of the lead in the research that's been done. And I think lastly, I'd just like to thank our MPS team, which is Vanessa, Anita, Anna, and Maria. So that's the end of the acknowledgements. Thank you. So Danielle, mic off now, yes?
Yes, it's Mike. I just wanted to remind the attendees to go ahead and, if you think of a question, write it in the chat box, and we'll answer them at the end. So now, Tom, take it away.
Okay, hello everybody. It's extremely nice to be here, and thank you, Daniele, for organizing this and our colleagues at RTI. And I'm really enjoying seeing all the people tuning in from around the world, so we can sit together at our desks and have this kind of a chat. I'm going to follow on with kind of a practical application component to much of the theoretical and background population genetic work that Ken and Chris did such a great job of overviewing. Work out the controls here. Yeah. So let me mention just a little bit about the International Commission on Missing Persons. Not everybody might know who we are and what it is we do, but we're now an international organization that's focused exclusively on problems of the missing, and we have a very strong forensic component of that, spanning a number of disciplines, but very importantly including DNA identification. So around 2001 or so, it began to be apparent that one could, using highly optimized methods, get STR profiles out of highly degraded bone samples with a certain success rate. And the ICMP was established to work in the countries of the former Yugoslavia to deal with some 40,000 missing persons, most of whom were present in or recovered from mass graves. And we went on to very intensively optimize the laboratory throughput mechanisms, the low copy number validation, and importantly, the computer-aided or computer-driven DNA matching for making kinship analyses. So we came into a method of a very high throughput manner, recovering STI profiles from these degraded bone samples from mass graves and other contexts, and comparing them to family members of the missing. And so this is now jumping forward quite a number of years in time, but this indicates where we are with the process. We have family reference samples representing about 30,000 missing people. And you'll note here that we have multiple family reference samples per missing person. So it works out to be about 3 or so per missing person. So we have to work very hard in public outreach to achieve that degree of collection, find close first and second degree relatives, or in some instances more distant relatives. But don't have I'm not going to go into this slide in any great detail, except to mention that in that particular work context alone, we've identified over 18,000 people using highly certain DNA analytical procedures.